blood. And take our notes on blood. Throughout this, there will be little activities for you to do where you have to stop and that type of thing. I'm only going to assign bits and pieces of the notes as we go along so they won't be overwhelming for you to do them all at once. It takes a little while to get through this section, so we are going to work on it the best we can. Hopefully we'll be able to get some activities and labs, etc. together. So when we're talking about blood, it goes under an overall arch-reaching category called serology. And serology not only covers blood, but it covers any bodily fluid that comes out of any orifices of our body. So we all know, or if you don't, you will know now, that an orifice is an opening. So any fluid that comes out of any opening of your body is considered a bodily fluid and falls under the category of serology. So we're talking about tears, we're talking about saliva, we're talking about blood, we're talking about vaginal fluids, we're talking about any type of rectal fluids, anything that would come out of an opening of the body. So this has to urine. This has to do with a lot of things. Serology has really started to boom since the 1950s, and the most important procedures came later on in the late 80s, 90s, but especially in the 2000s with DNA techniques. So because of the significance of DNA and what it can tell us and how it can narrow down our gene pool, our suspect pool, is the reason that we put so much money into it. As forensic scientists, our number one job is to figure out who our suspects are. And every test we run, we only run it to narrow down our suspect pool. So every time we talk about a technique that we're using here with serology, the main purpose of it is to take our suspect pool from being very large to making it smaller and smaller and smaller. Thus, that's why DNA is so important, because DNA actually is what we call individualized evidence, which brings it down to one suspect. And that's thus the importance of DNA testing. So when we talk about the blood, there are three main characteristics of blood. It is made up of plasma, and that is what makes up the biggest portion of it. This is the fluid part of your blood. It is secondly made up of cells, and thirdly made up of serum. Now, there are three types of cells within our blood, and the first one are our red blood cells, erythrocytes, and their job is to take the oxygen and carry it all around the body. The second type of cells that are in the blood are what we call our leukocytes, or our white blood cells, and they are responsible for helping our immune system. What they do is they clean out things in the body that aren't supposed to be there. So if you have an infection, these white blood cells go there to fight those off. The third type of cell that is in our body is what we call thrombocytes, and these are platelets, and their job is to allow our blood to clot so that if we get cut, etc., we don't bleed to death. Now, the third characteristic is serum, and that is the very liquid in there that separates the blood when the clot is formed. So once that clot is formed, then we get a little bit of fluid called serum and then blood. So it's a little bit thicker than the blood itself. Now, historically, we started doing something called blood typing, which will be the first type of lab that we do, blood typing, in the 1950s. And that what they did then discover there was 4 types of human blood. And we base what type of blood you have, or blood type that you have, based on specific antigens. Now, we know from biology, if you've taken it already, that antigens are types of proteins, and these particular antigens are found on the surface of red blood cells. And then in the 1940s, what they found was that after they had established the blood antigens A, B, they then, in the 1940s, realized that there was yet another type of antigen, which we call the Rh factor. They call it the Rh factor because it was actually discovered when they were doing research on rhesus monkeys. Now, when we talk about why do we use blood typing in forensics, we use blood typing in forensics because what it does is it narrows down our gene pool. There's only a certain percentage of people that have A blood, certain percentage that have AB blood, certain percentage that have O blood, and certain percentage that only have B blood. But if you also notice on the slide here that the Rh factor really narrows it down because when you look at this, Rh positive in Caucasians, 85% are positive. So that means 15% of Caucasians are Rh negative. Only 6% of Black Americans are Rh negative, and only 1% of Asians are Rh negative. So you're taking your gene pool from 100% and really being able to narrow it down. And that's one of the reasons that they use blood typing. Blood typing is also very, very cheap to use, therefore, to do, and it can be done on site very quickly. We're just going to review very quickly some of the words and the terms that we're going to use in this unit. ABO groups, all that does is it's based on whether we have A, B, or both antigens. The Rh factor can either be present or not present on red blood cells. You put a plus if it's present, and a negative if it's not. The antigen is that protein or that substance, and what it does is it stimulates the body to bring an antigen. So these are found in the plasma of the red blood cells, in the membrane, and it determines the blood type. The antibody that is attracted to it, and again, this is part of our immune system and how our body functions in many, many areas, is we have something that attracts this antibody. And so when that antigen is present, an antibody is then reacted to react to it. It's really the basis for our immune system, which we talk a lot about in infectious disease. Agglutination is when our red blood cells clump together, and this gives us coagulation. This is how we test for antibodies. So we'll get this clumping together with different types of antigens when they're mixed together. And then based on what we're mixing with, we can tell what type of blood type it is. So when we do our blood typing lab, that's what we'll be looking at. So blood typing is, if I have blood type A, then I have antigen A on the surface of my blood. And what will happen is if blood A and B are mixed together, we'll get that coagulation that will happen. Because remember the definition of agglutination, it's when the two different types of antigens come together, they'll clump together. If I have blood type B, that means I have antigen B on my red blood cells. So if I mix, again, A and B together, I'm going to get that clumping or that agglutination. Now, if I have blood type A and B, that means I have both of those antigens on the surface, and I won't get any clumping with either type, A or B blood. So if I have A and B blood type, and I mix it with A blood, won't get any clumping. Mix it with B, won't get any clumping. Blood type O has no antigens on it, neither A or B, and it's not going to coagulate with any of those. So when we do our red blood typing, we're going to have what are called anti-factors, and we're going to put those in, and we're going to essentially be mixing them to see if it reacts or doesn't react. This is video here I'd like you to watch while you go through this slide. I'm going to skip through it, however. Now, these are our blood types right here. I have type AB, AB, and O. Now, on type A blood, the antigen present is A. What it does is it attracts the antibody B. The people with type A blood can give to A and AB people, but they can't give it to B because remember, that means it will clump. It can get blood from O because there's no antigens on it, and A because it won't clump. B blood has a B antigen, A antibody, and you can get blood from B because it won't coagulate, and you can get blood from AB because remember we said AB won't clump together no matter what it's mixed with. It can get blood from O and B because if that goes in, it won't clot. Notice then it can get blood type The one that is in every single category is O. That is your universal donor. Universal donor. It can be given to anybody and there will be no clotting. That's important because if you're in a car accident, that people don't know what your blood type are, then you need blood in the accident. They will give you O so it doesn't coagulate. So if I have blood type AB, my antigens are A and B. Neither antibody is going to come at it. You can give blood to A and B, and you can get blood from anybody. So blood type AB is your universal receiver. If you have AB blood, you get blood from anybody and you're okay. If you have type O blood, they will call you a lot because they're going to want you to donate blood because you are the universal donor. But you don't have any antigens on. It attracts both antibodies A and B. This 1B should be up in the slide above it. It can give blood to everybody, but it can only get blood from other O donors because it will coagulate if A or B is put in there or AB. So those are our blood groups, and this is how it is broke down. 45% of the population have O blood, 40% have A, 11B, and 4% have AB. So the smallest population of blood percentage that people have for forensic purposes is AB blood. So again, this is how blood typing will narrow down your suspect pool, which is what we want to do in forensic science. And I apologize for the movement of some of these slide numbers. When I transferred it over to use the Peer that's what happened. All right, so here I just want you to go through and do this little fill in the blank as kind of a double check for you. So when you're doing these and you get to this slide, do that. Another thing we look at blood for, we'll do a lot of this here, and I'll be sending some blood home with you to be able to do this, is we're going to look at a stain at a scene, and it can tell us a lot of things. First of all, we want to figure out, is it blood or not blood? Is it food? Is it something else? Because when blood dries, it can look very, very different, though it takes about 3 days for it to completely dry on a surface. It's very sticky. Once we figure out or determine is it blood or not, we want to determine is it human or not. We don't want to send out testing to labs that are very far behind in their testing and are very expensive to use, and then to come back and find out it's the blood of a German Shepherd or, a neighbor cat, right? So we have to first then determine is it human before we do anything else. We can figure out maybe whose is it. From that blood, I can determine what is the blood type, is there any alcohol or drugs present, and we can figure out how was that blood put there. Was it from a low, medium, or high velocity? Was it from a knife? Was it from a smear? Was it transferred from somewhere else? All of that comes together to feed the big question and the mystery of who did this, what happened, right? You never come up with a predetermined scenario. You let your evidence give you what that scenario is. So first thing on that list was to determine is it blood or not. So we're going to practice doing some different types of tests to determine is the blood present or not. These are called presumptive tests. And in presumptive tests, it's going to presume that that's what we're testing and that's what we're getting. So the Kastle-Meyer, the color test, is a test that will turn deep pink if blood is present. There are also some tablets that we can use called hematest tablets, and they turn it a blue-green. We don't use those. They're very, very, very expensive. Kastle-Meyer does just as good of a job, and it's much cheaper. So that's what the test that we will be doing in class. The luminol test reacts with blood and it produces a light, and so you can see it in the black light. It's what you see on all the CSI shows. It has to be very dark. It doesn't always give you the big aha moment that it does in the CSI shows. And one of those reasons is I can't smear real blood all over the walls and then wipe it up and then use the reaction. most of the blood, all the blood I work with is simulated for obvious reasons. So it's not the same aha moment, but we will do that procedure. We'll do it on cloth that has blood on it to see what that looks like at a much smaller scale. And plus, it's very, very finicky. It the the chemicals only last about a day to do that. So another thing we do to tell if is the blood animal or human blood is we can look at it under the microscope, um, and that is very easy to do, and it looks very different. Animal blood, number one, the red blood cells have a nucleus in them, so they look very different. They're also very large. When you look at the human blood right here, they're very, very tiny, so they're much, much smaller, and there is no nucleus inside red blood cells. So you can see in this line that most of these cells here are red blood cells. The ones with the dark spots in the middle, those are white blood cells because white blood cells do have a nucleus, so you can see those in there. So if you use the microscope method, it is very, very easy to tell the difference between human blood and animal blood. Another way you can tell the difference is what we call a precipitin test. And in a precipitin test, what you get is a coagulation. You get a precipitate, right? And what happens is it will react with human proteins if human blood is present, right? And you'll get this precipitate. It only takes a small amount of blood, and you can test it right then and there on site, which is good. So here what happens. So I have, say, blood is injected into a rabbit, and then we get the antibodies that are from that, right? The rabbit's blood is taken out and placed on a sample of blood. So we have animal antibodies, and we're placing on a sample blood. The sample will react with human proteins if the human blood is present. Okay? So if it coagulates, then we have our answer there. Okay? It's very sensitive, very quick to do. So that's how we figure out, are we human versus animal, which is one of the things we want to figure out. The other thing we determined was we could figure out, where did the blood come from? How did the blood get that? And we use that by determining blood spatter evidence. So you can tell there's a lot going on here in this chapter. So it's going to take us a while to get through this, especially because there's going to be times I'm going to want to send stuff home so that you can do some stuff at home. So forensics scientists will go to a crime scene, and their sole job will be looking at, where is the blood? Because of the law of physics, how did that blood get there? What came first? What came next? You can write a whole scenario of that. It is a whole science within itself in forensics. You can look at the characteristics of blood drops, and what we do is we look at, is it the shape of it, right? It will stay very spherical if it collides with the surface. Once it impacts the surface, you'll get the stain. If it falls from the same height, the same surface, the same angle, you'll have the same shape. So in other words, if I drop 5 drops from 3 foot, they're going to have the same, approximately the same shape, same size, right? If I change that height, you're going to have different sizes. Still going to be the same shape because I'm coming from the same angle. The shape differs when you change the angle that the blood impacts at, right? Because most blood just doesn't go straight, right? Somebody gets shot in the chest, the blood's not just dripping straight down. It's going from a certain angle onto the next area. So we're going to figure out, we're going to design a lab on how can I tell the shape of blood as the height increases or decreases. The amount of blood is going to determine the blood droplet size too. So when we do our labs, we really want to make sure that we just put a certain amount of blood down at a time, because it will make a big difference on what the size of that is. Now, the size of the blood droplet that's come out is going to be completely dependent on um, where it comes from in your body. So your blood droplets that are like pouring out of your nose from a nosebleed may be very, very different from, something coming out of the tears of your eyes. The area that the blood hits is what we call the impact area, right? We call that the target. So whenever we're talking about the target, it's where the blood fell. And that will also impact the shape of the blood because of the how porous that may be or not porous it may be. So to review what we just said, what is it that affects the shape of a blood drop? First, it's the size. Second, it's the angle that it comes at. Third is the velocity, right? So is it coming from a slower speed, a higher speed, a medium speed, that type of thing? And that depends on what's causing the blood to come out, the height that the blood is coming from, and the texture of the target. So if it's clean glass plastic, it's going to be smooth edges. If it's on rough, it's going to have a little scallop on the edges, kind of like, concrete is very porous. So it's not going to be a beautifully shaped circle because of that. So what are the questions that we can answer by blood spatter? And we talked a little bit about this. We can tell the distance between where did the blood come from, where did it land? What was the point of origin of the blood? Where did that blood start from? So where was the victim when that happened? What direction did it move in? Um, um, and the number of times that victim was shot, hit, and how many of those times caused the bloodshed or the dispersal of that blood at a crime scene. The type and direction of the impact. So was it from a gun? Was it from a baseball bat? From a knife? What was that? And at what velocity was it? The position of the victim and the position of the perpetrator. And how did the victim move? If they were shot 3 times, they weren't all in the same position each time, because if you get hit, or even if they were hit, they're going to move in a certain direction for a certain amount of space. Again, some terminology that we'll need to have and understand for the serology unit is number one, angle of impact. That's what angle did it strike the target at, right? Did it strike it at 90 degrees, 10 degrees, etc.? Was the blood stain transfer? So when it comes into contact with something and it leaves a pattern of an image on the surface, that is a blood stain transfer. Back spatter is the blood that is directed back towards the source of the energy. Well, so in other words, especially if you are using something of a high velocity like a gun, you're going to get some back spatter. So the blood from the victim isn't just going to go towards the target away from the weapon, it's going to come back at you, and that's called back spatter. Cast off is the blood that is from an object in motion. So that, typical knife stabbing. You stab something, and as you're moving the knife up and down, up and down, up and down, you're going to get what's called cast off from that. And we'll do another lab where we actually demonstrate that. So there's a lot of labs that are going to be done in this, and we'll be able to do them hopefully maybe in class one day, and if not, I'm going to send blood samples, fake blood home with you guys, so you can do some of it at home with your, and we'll do some here as demonstrations too, whichever works out the best. Another is a contact stain. That is when I have 2 wet surfaces of blood touch each other, and then it kind of gives you a third thing. It can be transferred from something that was, you don't know what object it came from, but it came from another source. A swipe is when it is transferred to a surface that did not have blood on it. So you had blood and then you move something across a clean surface. A wipe is when something that doesn't have blood on it then moves across a surface that has blood on it. Directionality, and we're going to talk a lot about this, and that's the direction of the blood travels in space. from the point of origin. So it's going up, down, away from the victim, towards the victim, etc. Terminal velocity, this is the greatest speed that a free-falling blood drop can accelerate in air. Um, it is dependent on the acceleration of gravity, which we know doesn't change, the friction of the air against the blood. So it's approximately 21.1 feet per second. Now, when we're talking about high velocity blood spatter, it's usually greater than 25 feet per second. It's usually around 100 feet per second that we get a fine mist appearance. And we're going to do samples of low, medium, and high blood spatter so you can see these differences. Medium velocity is 5 to 25 feet per second, and low velocity is 5 feet per second. Now, all of these is still fairly quick, right? If you had to go 5 feet in 1 second, you're moving at a pretty quick pace. When we look at blood stain, we can have different shapes of our blood. One is round, it falls straight down at 90 degrees, and we can also have elliptical shapes. And these blood drops, they become longer the further you get from 90 degrees and the closer you get to zero. So that angle can be determined by looking at the width and the length and using the inverse sine. Don't get hung up on this math, please. The beauty of forensics is that we use a lot of math to help determine things, but it's math that makes sense, so it's not crazy math and you're like, oh, I hate doing these stupid word problems. It is pretty basic math. I don't ask you to use your calculator for the inverse sine. I give you an inverse sine table. Basically, the only thing you have to do is determine your width divided by your length. And we'll talk about how to measure all that out, etc. So here's the angle of impact. So the blood drop on the left is at 90 degrees. It's nice and straight. You'll notice that there's some little spikes that come off of those, and that's what we call those, is spikes. The higher up you get, the more spikes you get. Even the higher than that, you'll get what's called satellites. And those are specks of blood that come off that are like satellites or little bits of blood that are around that blood drop, just like satellites are around our planet. The one on the right is the elliptical one, okay? So the one on the left, between 80 and 90 degrees, the closer to 90, the more round it is. And at about 30, it starts to produce this little tail down at the bottom. And the more acute the angle, the easier it is to determine the direction of travel, because this tail is going to help us tell us what direction the blood is traveling in. If we go opposite of that, we can tell where the victim was. So the harder the surface and the less porous, you're going to get the blood drop's not going to break apart as much. The softer and more porous it is, it starts to break apart. You can tell the difference between the left side and the right. The pointed end of the blood stain faces the direction of travel. So the tail is the area that it's traveling to, okay? The tail is the area that it's traveling to, okay? So the pointed end of the blood stain faces the direction of travel. The direction it's going to. We are going to do a section in here called the area of point of convergence. And what this does is we can, by determining the points of convergence of blood spatter, determine a number of things. We can determine where the blows occurred, which blows occurred first, and how many of them occurred. Again, this is going to be simple geometry, and we map it out on a wall. We look at some blood drops, we map them out on a wall. And what we're doing is we're going to draw lines away from the tail, right? Because we want to go to the point of what we're looking at. We want to find the point of origin. So we want to find where was the victim. We know the tail is direction of where the blood's going, which means the victim is opposite. So you always draw lines through the blood drops and find a point of convergence. When we go to do this, I will give you explicit directions. We'll practice it and do all that before we do the lab. So blood evidence is what we call class evidence. There's two types of evidence in forensic science. First, we have individual evidence, which narrows down to one piece of evidence or one individual. All right? And that's the kind of evidence we really like, because then we can say this piece of evidence was definitely at the crime scene. This was the definite gun that was used there, that type of thing. DNA, individual evidence, because I know it was that person. But class evidence is really just as important. It's things like blood typing, blood spatter, that type of thing. What it does is it narrows down the suspect pool. It narrows down the pieces of evidence, and it makes things so we can start figuring out what our scenario is. Blood stain patterns are circumstantial evidence in the court of law, and you will find that in most instances, circumstantial evidence is the evidence that is really used most in court. Experts would say that many points, including direction of travel, height of the perpetrator, position of the victim, left and right handedness, and whether the body was moved or not, they could argue those points, saying not necessarily circumstantial, but more class evidence. But those are things that help us put the story together. Now, there are things called secretors, and 80% of the populations are what we call secretors. Now, remember, blood type antigens, we said, are found on the outside of red blood cells, right? And that is true. But they can also be found in very, very small amounts, but high enough concentration amounts for us to be able to test in bodily fluids in 80% of the population. Saliva, semen, vaginal secretions, gastric juices, all of those things we could, in 80% of the population, determine blood type from without having a sample of blood there, which actually is really an important thing. Now, after this slide is actually a quiz that I'm going to have you take when we get to this portion, and you're going to submit this. I really want you to take your time with it. You can look over your notes. You can do that type of thing, and I will be taking this as a grade. And if you want any more information about serology, this is it. So that is the notes for this unit as of right now.